Good morning, everybody. Welcome to Pediatric Grand Rounds. We're so excited for those of you who are here in the room with us today in Wright Auditorium and all of you who are here with us on WebEx today. As usual, if you have a question and you are online, feel free to type it in the chat and I will be monitoring the chat and ask your question at the end of the presentation. I'm so excited today to introduce Dr. Tommy Wood, who is an assistant professor of pediatrics and neuroscience at the University of Washington, where his laboratory focuses on brain health across the lifespan. This includes babies born preterm, adults who experience brain trauma, and the confluence of factors that can affect long-term cognitive function and risk of dementia. He received a bachelor's degree in biochemistry from the University of Cambridge, a medical degree from the University of Oxford, and a PhD in physiology and neuroscience from the University of Oslo. He's published dozens of scientific papers and lectured all over the world about brain health, metabolism, physical activity, and human performance. He's also spent more than a decade working as a performance consultant to professional athletes in multiple sports, including several Olympians and world champions. Alongside his work in medicine and research, he's worked with several digital health and performance companies to develop easily accessible methods with which to track human health and longevity. So excited to learn about this, so I'm going to turn things over to Dr. Wood. Thank you so much.
Thanks so much for having me. I'm really excited to be here with you and share some of our work in the Neonatal Neuroscience lab and beyond. One very brief update to the bio is I got promoted last week. So thank you. You already gave my background, so I'll just give a little bit of this because I think it gives some context to what I'm going to talk about today. So, like Suzanne said, I got my undergraduate degree in biochemistry from the University of Cambridge. I then studied medicine at the University of Oxford. I did my early postgraduate medical training at Guy's and St Thomas's Hospital in London, which is most famous for being right opposite Big Ben down in central London. And then before I sort of finished my training and specialized, I moved to the University of Oslo to do my PhD with Marianne Thoresen, who's one of the big names in neonatal neuroprotection. And so this is where I moved from being a human doctor to being a rat doctor. Before coming here to the University of Washington and doing a postdoc with Sunny Jewell, who's the former division head of neonatology, is now the director of the Institute on Human Development and Disability here. And with Sunny, I became a ferret doctor, and you'll hear about that as well. Then alongside that, as we've grown the lab and the work that we try to do, I've done some work analyzing data from large clinical data sets in the division, such as some of these large clinical trials that Sunny ran, the Peanut trial and the HEAL trial, and you'll hear a little bit about those as well. So I'll mention some of the additional things that I enjoy doing that I'll try and tie together in this talk. So when I was in medical school, I was head coach of the medical school boat club. We had one of the most competitive female rowing teams at the university, including several national level and international level athletes. When I was doing my PhD, I helped build a digital health company that worked with athletes on health and performance. This is where we started to work with athletes of all levels, including Olympians and world champions. Through that, I got a gig consulting with Formula One drivers through a company called Hintsa Performance. And I won't talk about that much today, but if you're interested, there was a nice article in Forbes that came out like a month ago where they interviewed me on some of that stuff. And then, being interested in athletes and the brain, I do some work in military and athletic groups trying to prevent and treat traumatic brain injury. I'm also a founding director and trustee and current treasurer of the British Society of Lifestyle Medicine, which is the fastest growing lifestyle medicine society in the world. And I'm on the scientific advisory board of this dementia prevention charity in the UK, Food for the Brain. I'll tell you about that right at the end. So I appreciate this is a pediatrics grand round, so we are going to talk about babies. And these are the two main groups that we study and try and improve the treatment of in the lab, in the Neonatal Neuroscience lab. The first is babies born preterm. As most people here know, that's around 10% of live births in the US. The more preterm you are, the more likely you are to either die or suffer some significant neurodevelopmental disability or impairment. And currently there are no targeted neuroprotective therapies available for babies born preterm, particularly those extremely preterm. The other group that we study are those with hypoxic ischemic encephalopathy. These are treated in high-income settings, high-resource settings with therapeutic hypothermia. We have our co-director of the NeuroNICU here. And however, this is not a therapy that works in low-resource settings, and still about a third, at least, of babies treated with hypothermia have poor outcomes, so we're still looking for adjunctive therapies. As I navigate all the work that we do and things I'm interested in, these are some guiding questions that I have. I'm not here to tell you that I have the answer to these questions. These are just, I think, important questions that we need to have as we do basic and translational research. The first is, are we using the right models? The second is, how can we better leverage common disease physiology for better treatment and prognosis? Are we investigating and intervening at the right scale? We increasingly focus deeper and deeper and deeper into the cell in neuroscience, and honestly, that hasn't really gotten us anywhere. And then can we gain additional insights by ignoring traditional silos and looking across multiple fields? So we'll start with HIE and the models that we use to study that. Historically, and until very recently, we could say that neonatal neuroscience is world leading in the development of therapies. It has a very well-developed pipeline of animal models, starting in vitro and ex vivo, then rodent animal models, and several large animal models, the fetal sheep, piglet, non-human primate. And this is the pipeline that was used to develop therapeutic hypothermia for HIE through the 80s and 90s and into the early 2000s. And in reality, this is the last big major breakthrough in neurology in terms of treating a condition that had developing a treatment that had a very significant reduction on outcomes. There hasn't really been anything that had this big of an impact since then, and this was more than 20 years ago. However, there's a large gap between the clinical and preclinical efficacy of therapeutic hypothermia. It works really well in the lab, and it works kind of well in the real world. And again, it's not beneficial in lower income settings, which is where the majority of HIE occurs. When I did my PhD, I did a lot of work on hypothermia in this rat model of HIE. This is the Vannucci model. And the way the Vannucci model works is you ligate the left common carotid artery, as you see on the left there. Then you put the rats into this chamber and expose them to hypoxia. This is our little rat hotel, as we called it. Each individual animal has its own gas supply. We then randomized by weight and sex and litter to either normothermia or hypothermia. And in general, when we're screening therapies in this kind of model, because it's a unilateral injury, you can see we lose tissue on one side of the brain, we compare that to the other side of the brain, and we get this thing called area loss, and you can see on the left-hand side. And in general, historically, you see about a 50% reduction in the amount of injury that you see with hypothermia. When I was finishing up my PhD, I become kind of frustrated because hypothermia, was supposed to work really well, but actually across different experiments, it didn't always work. So one thing that we did was this big meta-analysis to try and figure out what is it that's affecting the variability. And there were lots of experimental variables that really seemed to matter, like the barometric pressure on the day and the size of the animals and all these different things. And so these tiny details really, really matter when you're doing this kind of work in animals in the lab. But one of the interesting things that came out of this was an apparent sex effect. So if you look at area loss there on the y-axis, and then you have our groups on the x-axis, in females on the left, you see a much bigger decrease in area loss in the hypothermia group, whereas in the males there was a reduction, but this was not significant. This sex effect is actually the thing that most people cite this paper for, and actually led many people to assume that hypothermia was less beneficial in male infants with HIE without ever seeing any evidence of that in humans. So this was something that was really important for me personally to answer, and it's something that we can hopefully answer very soon. We're just about to submit this paper. We did an individual patient data meta-analysis of the 5 original cooling trials. These are the cooling trials that led to therapeutic hypothermia becoming standard of care. And doing this and adjusting for severity of HIE, you can see both the males at the top and the females at the bottom, there's an almost identical reduction in death or neurodevelopmental impairment at 2 years. So no sex effect in humans, despite there being one in rats. So this then brings up the question of whether we're using the right models. And here are some current problems in preclinical HIE research that we think about a lot as we try and develop new therapies or adjuncts to therapeutic hypothermia. The first is that what works in rodents rarely translates to humans, and that's actually the case for most acquired brain injuries as well as neurodegenerative conditions. The rodent models just haven't really translated that much to humans at all. And then most cases of HIE worldwide aren't suitable for hypothermia, and that's where most HIE occurs. It seems that it may even be detrimental. It may increase death, as it did in the Helix trial, which they did recently. And there's also a different pattern of injury, which may be part of it. So there's less deep gray matter injury, which you see in a sort of a typical acute asphyxial event in high-income settings. And there's more injury to the white matter, suggesting that there's a more prolonged or intermittent injury, as well as maybe being affected by a background of lower birth weight or nutrient insufficiency. So it's just a different injury, and we need to find a different way to model that. So this is where the ferret comes in, and this is a project that Sunny started more than 10 years ago, and we've since been working on in the lab. And what's interesting about the ferret is that it's the smallest animal with a gyrified brain, as you can see in the middle there, unlike the rat, which is completely smooth, it's lissencephalic. It also requires a more prolonged injury process to see significant injury, and that's something we've worked on, so maybe more similar to HIE in lower income or lower resource settings. And it's amenable to lots of long-term behavioral testing. It's sort of like this perfect middle ground between rodent models and larger animal models. So when we started this, we basically needed to start from scratch because there wasn't an injury model. Nobody had really done work in this area before. So this is just one of the first papers we published looking at the development of white matter in the ferret. You can see that on the left-hand side from postnatal day 5 up to adulthood. You can see the myelin basic protein development. So these really nice gyri, as well as that penetrating white matter, much more white matter in that brain than in the rat. And then we also had to test how do these animals develop. So on the right-hand side, you can see this behavioral score that we developed. And there's sort of a relatively young ferret on the left at the top. And then by the time they get to sort of 6 or 7 weeks old, you can see them there wrestling with their littermates. We then developed this model, which is what we use now. And it's much more complex than what we do in the rat, but we give an inflammatory stimulus, LPS, lipopolysaccharide. We then do bilateral carotid artery ligation. One side is permanent, the other side is reversible. We expose them to intermittent hypoxia and hyperoxia. We then reverse the ligation on the other side, like a reperfusion type injury. And then over time, we can look at things like reflex development, how they perform behavioral tests like walking in the catwalk, and then we can do MRI and look at white matter tracts. So when we first developed this model, we got a grant. Sunny got a grant from the Gates Foundation to try and test some therapies that might be useful in lower resource settings. So this was our first published study. These were our main treatment groups. So we had control, we had a standard saline vehicle in the injury group. We were testing uridine monophosphate. They were very interested in uridine as a very accessible therapy. Erythropoietin, which is something the lab has lots of experience with. It's a very promising monotherapy for HIE, particularly in lower resource settings. And then hypothermia as the kind of standard. And you can see on the left-hand side, this is a control brain. You can see all those gyri, those very nice long sulci. In the vehicle group, you start to see narrowing of the gyri, so you've created some cortical injury, as well as the sulci become more shallow. In hypothermia, we didn't really see an effect, and I'll show you more data on that in a second, but EPO seemed to reverse some of these effects of the injury. And here's just some graphs of that, and what we saw was really that the model responded as we expected it to if it was going to be modeling the kind of injury that we see in HIE in lower resource settings. EPO was significantly neuroprotective. You can see that in the purple with our pathology score on the left there. Uridine actually was not beneficial, unfortunately, and hypothermia also was not beneficial. Again, similar to what we see in lower resource settings. So we have this model. We think it's maybe going to be useful to test new therapies potentially the majority of infants who experience HIE, but how do we even pick what therapies we're going to put into this model? And this is now a pipeline that we've started to develop in collaboration with Elizabeth Nance, who's an associate professor in the Department of Chemical Engineering, using a slice culture model where we take live brain slices from ferrets, we do an injury model, and then we can look at all the different regions of the brain, how they respond to injury and how they respond to therapies, and this allows us to sort of screen therapies in a high throughput manner. And that's even more important because we're now starting to see that different areas of the brain respond differently to different therapies. So we're probably going to need a combination or cocktail of therapies, and then that expands the potential number of treatments even further. So this was the first paper we published using these ferret brain slices. We take these slices, we culture them for 3 days, and then we expose them to oxygen glucose deprivation, which is like that hypoxic ischemic event that we think the babies are experiencing. And you can see just in 3 regions of the brain, the corpus callosum, the hippocampus, and the cortex, the amount of injury that you see is very different. And then we have 2 therapies here. We have erythropoietin again, and we have acetoacetate, which is a ketone body. And depending on the region, you see different responses both to the injury and the treatment. We also saw that where we're seeing injury is very similar to what we're seeing in the in vivo model that I showed you before. So hopefully what works in the slices will then work in that model. So we can look at various things in this model. At the top here, this is looking at the structure of mitochondria. You have the control group, the injury group, then you have EPO and acetoacetate, and you can see after the injury, you get this sort of fission of mitochondria. You get complete dispersal of the signal. This seems to then be improved a little bit by both acetoacetate and EPO. We're also particularly interested in microglia, which are the main resident immune cells of the brain, and so they're stained with this stain IBA1 that you can see at the bottom. And so again, we have 2 regions of the brain, the cortex and the hippocampus. And you can see across the groups, even in the control, the microglia look different in different regions. And then you lose some microglia with the injury, and that seems to be restored by the therapy, at least here, particularly by EPO. What we can then do is we can really dig into what these microglia look like. So we have this pipeline, again, developed by the Nance lab called VAMPIRE, which stands for Visual Aided Morphophenotyping Image Recognition. Vampire sounds much cooler. And what it does is it can trace the outside of all the cells and characterize them in different ways. So we can look at their perimeter, their area, how circular they are, which is thought to be a measure of how activated or pro-inflammatory they are. And again, you can see across multiple regions of the brain how these cells respond to injury is very different. So sometimes perimeter goes down and sometimes it goes up, and that's very dependent on where in the brain we're looking. So again, just suggesting that we can't just look in one area of the brain or assume that all the brain will respond the same. On the right-hand side, you can then see what this algorithm does is it separates out all the different microglia based on their different shape parameters using something called principal component analysis. And then it classifies them into these 5 different shape modes that you see in the middle. And what was really interesting in this paper was that seeing how microglia responded in one area of the brain then predicted whether that area of the brain would respond to a therapy. So there's this direct connection between how the brain responds to an injury and then whether we might be able to treat it with a therapy. So as we kind of develop this pipeline, we're now starting to plan testing of multiple treatment combinations in vitro, then moving the most promising ones in vivo. This allows us to screen multiple therapies and identify optimal combinations. We can then look at all the things like the transcriptome, the cellular responses like I've shown you. And then in vivo, we can do the sort of more translational outcomes like behavior, MRI, and neuropathology. We've largely focused on repurposed or approved therapies, so things like erythropoietin, azithromycin, both of those are already used in preterm infants, for instance, or supplements like melatonin, caffeine, vitamin E, vitamin D, things that babies are often given anyway. This gives us a faster route to translation if they're beneficial. And we have 2 current projects. So we have one funded R01 to do this pipeline for extremely preterm equivalent brain injury. And we just got what looks like fundable scores to do the same thing for a version of the model that looks at lower resource setting type injury. So this is kind of the pipeline that we're working with. And I'll show you just a little bit of what that might look like. So in the extremely preterm equivalent model, we tested azithromycin and EPO and their combination. And we've looked at this two different ways. One is how much LDH is released into the medium of the slices, and you can see that on the left there. And this is essentially how many cells have lysed and died in the brain. And you can see on the right, of that graph that azithromycin and EPO significantly reduced that compared to the OGD group, which is in blue, but both treatments individually didn't have that effect. We can also count dying cells in the brain, and that's what we have on the right-hand side. And you can see a significant effect of azithromycin then in purple compared to OGD, but an even greater effect with the combination therapy. When you have multiple drugs, you can then see, is there any evidence that these drugs are either additive or synergistic? So we applied lots of different ways of doing that. There are 4 different models here, and we've looked both globally with LDH and the pycnotic or dying cells, as well as within each region. And with each type of model of looking for synergy, you can kind of see a tick where there's evidence that these 2 drugs are synergistic. And so it does seem that azithromycin and EPO are showing synergy, which means that there's promise that they might be a good combination to use in the model. What's interesting is we can then look at how the brain is responding to the injury based on the transcriptome, so which genes are being upregulated or downregulated. And so on the left, you can see a volcano plot. And these are the genes that are upregulated and downregulated in response to injury. And then on the right-hand side, what we did is we looked at genes that were upregulated by injury, weren't affected by azithromycin or EPO individually, but were when they were applied in combination. And these top 2 genes here, HMGB1 and NLRP3, are 2 like really classical cell death pathways that seem to be being reversed when you give this combination of azithromycin and EPO, but not when you give them individually. So another interesting aspect here is that we couldn't predict that this would happen. You can only find the effect by actually testing it. So in some versions of the projects that we've gotten funded, we're not screening drugs separately and then putting them together. We're just going to test like 100 combinations and see what pops out. So we really have to be able to do it in a high throughput manner. So if we're then, hopefully in the process of trying to improve therapies, something else that's a big need in the field is biomarkers of injury to try and improve prognostication, clinical decision making, maybe stratification of infants to different treatment strategies once we have some of those. And what we've tended to find and what the field has tended to find is that complex biomarkers, generally, they perform very well maybe in one single site in one single small study, but then they don't have good external validity and they just don't work in other cohorts. And this has really been our experience as well. So as an example of that, these are data from a primate model of HIE that Sunny developed and ran for several years. And in this model, we were trying to look at a complex panel of cytokines to see whether these would predict long-term outcomes. In these primates, we had cerebral palsy and motor outcomes out to 6 months. And all the different little forest plots basically show what was significant or not at different time points after injury. And only the black boxes are the ones that were even significant after accounting for multiple testing. And so there were a couple of things that maybe popped out, but basically nothing predicted long-term outcome very well. And unfortunately, in some of the human trials, we've seen something similar. So in the peanut trial, again, there were a large number of, so this is extremely preterm infants who were randomized to either EPO or placebo. And a big subset of them had a big panel of cytokines several times after birth. And there were some slight correlations between some of the biomarkers and injury. So here's a tau concentration, and then looking at different Bailey scales of outcomes at 2 years. And there's a slight correlation there, but basically there was nothing that said this is a cutoff of tau that we can use to predict outcome, unfortunately. So one thing that we've become increasingly focused on is, instead of zooming into specific mechanisms, is starting to zoom out and just looking at more integrated physiological processes as biomarkers. And so this is one that we've published quite a lot on in the last couple of years, which is looking at thermoregulation after hypoxia ischemia. So we're back to babies with HIE, but something that we've known as a field for essentially 70 years now is that after acute asphyxia, babies become spontaneously hypothermic. So these are data published by Bernard and Cross, and you can see normal babies versus asphyxiated babies after birth. Core temperature drops in both after birth, but then the asphyxiated babies drop much further and recover much more slowly. Then a few years ago, we looked at this in the rat model that I showed you earlier, and we see essentially exactly the same thing. There's a significant drop in temperature after injury, and then this takes somewhere between 6 and 24 hours to recover. We, again, have shown the same thing in ferrets, and in both rats and ferrets, we see that the more significant the drop in temperature after the injury, the more significant the pathology later on, or the more significant the brain injury. So on the left-hand side, this is in rats. You can see the decrease in temperature after injury along the x-axis, and then the pathology score 7 days later on the y-axis, and you can see these are fairly linearly correlated. In the ferret, we found that those who became spontaneously hypothermic after the injury had significantly higher cortical pathology, as well as diffuse changes throughout the white matter, and this is now several weeks later. So within an hour of the injury, just measuring the core temperature of the animal seems to give us quite a predictive measure of how severe that injury is. And we've shown something very similar now in babies with HIE. So this was a secondary analysis of the optimizing cooling trial, where we've kind of had to flip the biomarker, because all these babies are cooled to 33.5 degrees Celsius, as is standard of care. But we can look at the temperature of the mattress that's required to get them to 33.5 degrees Celsius. So you can see that on the left-hand side. These are the mattress temperatures over time. And what's interesting is that in those with very severe injury that have the worst outcomes, their mattresses are warmer. It requires less active cooling to get them down to temperature, because they're already cold spontaneously. And actually, in some of the most severely affected infants, they have to be warmed up because they're so cold that they're below 33.5 spontaneously. What we then saw is that if we created trajectories of temperature management over time, we could see that infants whose mattresses stayed cool consistently had a 100% likelihood of survival without disability, and I don't think there's any biomarker that we have that can predict that, and that's what we see on the left-hand side. But when we're trying to predict more complex outcomes, unfortunately, things become much more complex. And so this is a paper where we were trying to predict outcomes on the Bailey scales of infant development from the peanut trial, again, so back to babies with extremely preterm brain injury or who were born extremely preterm. And on the left-hand side, you see a graphical network analysis. You'll see a couple of these throughout the rest of my talk. This is basically an approach where we have lots of different potential variables. We have one variable that we're interested in, so cognition score as a continuous outcome here. But we know that all these factors are also related to one another, so we want to try and adjust for all the different relationships in the network and then figure out what is statistically significant once we take all of that into account. The thickness of the line tells you how important a relationship is. If there is a line, that's statistically significant. And then the color of it tells you about the direction. So anything that's orange or red, that's a positive association. Anything that's kind of green or blue is a negative association. So we can look at all these different risk factors and predictors of poor long-term outcome in these babies. But what really stood out to me, and we saw this in multiple outcomes, is that the most important predictor, and you can see that on the right, they're sort of ranked all the predictions in terms of how well they predict the outcome. Maternal education was the most important predictor of cognition at 2 years in babies born extremely preterm. So that's after taking into account all the bad things that were in hospital, all the drugs that they were exposed to. Maternal education had the biggest effect. So this is where we have to start thinking about the scale at which we are investigating and potentially intervening in these complex neurological and neurodevelopmental disorders. And this is a figure taken from a paper that I published with Elizabeth Nance a few years ago. really talking about how we have to integrate multiple levels of physiology, biochemistry, as well as the whole organism and its environment to really understand whether we can translate something to improve outcomes. And related to that, this is a paper I published with Josh Turknett, who's a neurologist, colleague of mine, just a couple of years ago. And we kind of dug into this a little bit more to really give examples that people could understand, like why it's important to intervene at the right biological level. So here we have two different disorders, and then we have all the different levels at which we can describe them: the molecular, the cellular, the tissue, the organ, and the organismal or behavioral. And so in something like bacterial meningitis, we can measure the DNA damage, the pro-inflammatory cytokines. We can measure the physiological responses. We can measure the encephalopathy that comes from it and see the symptoms that come from it. But really where we need to intervene is at the tissue level by treating the meningococcus. And similarly, in sleep deprivation, we can measure all the things that happen to the sympathetic nervous system and cognitive function and cardiovascular function in those who are sleep deprived. And we could develop drugs to target those things. But in reality, what you need to do is spend more time asleep. And that's at the organismal, behavioral level. That's where we need to intervene. And it's obvious when I say it now, but this is what we do in most of neuroscience, is we focus on the molecular rather than the behavioral or the organismal. And so to kind of just hammer this point home, this is not our work, but very important related work done by Steve Miller's group, looking at infants again who were born preterm and whether they had brain injury, documented brain injury in the hospital, and then looking at their cognitive scores out to 4.5 years, as you can see on the bottom there. And what they found was that there was a significant interaction between the presence of injury and maternal level of education. And maternal education is a proxy for a whole bunch of societal and socioeconomic factors, of course. But what you see is that in mothers who had a postgraduate degree, if their baby had a documented brain injury, there was no effect of that brain injury on their long-term cognitive outcomes. But if with lower levels of education, you saw an increasing negative effect of that injury. So what this really tells us is that the environment that brain goes into is what's most critical for its long-term function. And now zooming forward several decades, we see the same thing in older adults. This is a paper that we published last year where we were looking at cognitive function score in older adults from the NHANES cohort, the National Health and Nutrition Examination Survey cohort here in the US. And we wanted to look at different ways that physical activity and other health-related factors affected cognition as this kind of idea of how we can look across the entire lifespan. And again, lots of very important things. Physical activity, very important. I'm going to talk about that a little bit more. But education was the most important predictor of cognitive function. So how we're stimulating that brain and then what comes with that as part of that environment seems to be most critical. And this is something that we see in other studies as well. So this is my colleague Yintai Yu, who's also on the board of the Dementia charity with me. And he and his team did this very nice meta-analysis looking at the different risk factors for cognitive decline across the lifespan and their magnitude of their effect. So you can see that on the y-axis, anything that's above the dotted line has a negative effect, anything below has a positive effect. And you can see that more schooling early in life, so more education, and then more cognitive activity late in life are the two most important positive factors for maintaining cognitive function late into life. So cognitive stimulus really being this big driver of cognitive function. And we know that if we lose inputs to the brain, we also lose function. So we see the other side. We see that stimulus is beneficial, and we see that loss of input is detrimental. So there's an increased risk of dementia in those that lose a sense, such as hearing or sight. And this is a nice paper that looked at cataracts and cataract surgery. So we see an increased risk of dementia in those with cataracts. But if you have cataract surgery, you see a significant reversal of that risk. And that reversal is on the same magnitude of an additional 4 years of education. So really important to keep that stimulus going into the brain. We also know that degree of social engagement and support predicts dementia risk, and then complexity and novelty of work decreases dementia risk. So the more complex and novel and different kind of tasks you have to do as part of your job, the more benefit you get for your brain. And then, unfortunately, the time of most rapid cognitive decline is at retirement because we remove that daily stimulus. Stimulus is also important when we're trying to recover from brain injury. So this is a systematic review that was done by a medical student in the lab, Marsaki Rhodes. It's just been submitted. And what he was really interested in looking at was interventions in the chronic phase of traumatic brain injury, where all sort of guidelines and recommendations kind of start to drop off. And he wanted to see, could we match specific interventions to specific symptoms? Because there are several common symptoms and different people experience different ones. And he compared them to the two main standards of care for traumatic brain injury in this setting, which is either physical therapy, you can see those two at the top, or just rest. And looking at these different symptoms, he then looked at whether they were different or not, and then the level of evidence, which is sort of hashed out. But what's really interesting is that the most beneficial sort of novel interventions seem to have some aspect of stimulus to them. So aerobic exercise, Tai Chi, and then particularly for cognitive symptoms, augmented or virtual reality, where you're having to do complex movements, but sort of you can do it in the safety of your own home in a VR setting. So these types of stimulus seem to be really important for the brain as it's trying to recover. So then as we try and apply this to ourselves, we might think, how does cognitive function change across the lifespan? And you might imagine a curve like this. This is sort of like an average, some kind of cognitive function on the y-axis there, age on the x-axis. And we know that early life, education, other skill development, this drives an increase in cognitive function that seems to peak sometime around the end of formal education. And then from there, it's just this precipitous, inexorable decline that we can't do anything about, except for maybe make it worse when we get stressed, when we sleep badly, or when we have a few too many cocktails. We then get this drop, and all we're doing is just making it worse, and there's nothing we can do about it. This is kind of how we think about the brain, right? But in reality, this maps onto how we use the brain through our life. So I've created this graph. This has sort of nervous system demands or stimulus in arbitrary units. And if we think about as the brain is learning and developing, learning to walk, learning to talk, learning social interaction, these are incredibly complex tasks that drive a huge amount of stimulus in the brain, and this is when we're improving cognitive function. We then get a little older, we learn to drive, we go to medical school, we learn the Krebs cycle, and it's hard, it's a little boring, but it's not as hard as learning how to walk. We're not driving that same kind of stimulus in the brain. And then we go to work and we do the same thing again and again and again and again. And we feel busy, but that's not the same as sort of driving this stimulus demand in the brain. Then we retire, we lose that bit of stimulus that we have. And then we think, well, I have to use my brain, so I'll do some Sudoku. And it goes up a little bit, but actually it's interesting that Sudoku and crosswords, they seem to have more of an effect on the brain similar to meditation and mindfulness than being this direct cognitive stimulus. So in reality, there's this big discrepancy between how we use our brain early in life and how we use it late in life. And then we argue that aging causes a decline in function, but it could just be that we're not using our brain anymore, and then function declines as a result. Just like if we stop exercising, we get less fit, and it's not because we got older, it's because we stopped exercising. So this is a graph from one of my favorite papers, this monograph by Herzog et al., which talked about how each of us have this really wide potential for cognition. We don't have this one single line that we follow over time. We have this really broad range that we can manipulate and change at any time. And the things that they talked about being really important were these three main groups. So enhancing the function of the neural substrate. This is things like nutrition, physical exercise, supporting the health of the brain. But then a big one being the level of cognition itself. So how we use and create stimulus for our brains, things like skill development. And then also the larger context in which an individual thinks, learns, and remembers. So you've learned these things, and you have to go out and use those in the world. And it's the process of using those skills that we've developed that then also maintains function. And then this becomes important, going back to the beginning of the story, because adults who are born preterm have an increased risk of later in life cognitive decline and dementia. This has been done in several studies. Particularly, there are large cohort studies in Sweden where they've looked at this, and they see with both those born preterm and with growth restriction, they have an increased risk of dementia and cognitive impairment. And then looking at younger adults who are born preterm, so maybe in their 20s and 30s, they tend to perform less well on cognitive function tests, and this is related to the degree of prematurity and the severity of illness in hospital, and then of course also things like socioeconomic status. But then interestingly, you see that in adults born preterm, they have accelerated brain aging, which you can measure on an MRI scan. So they seem to be particularly at risk for this kind of decline in cognitive function due to being born preterm. So then you might think, well, are there ways that we can intervene later in life if we're trying to improve the environment that this brain exists in? And as many of you, if not everybody knows, preterm birth is particularly associated with white matter injury. Especially the cholinergic white matter tracts that are important for things like processing speed and executive function and memory encoding. And these same white matter regions are also affected in Alzheimer's disease. So this is a part of a table from a paper that we just published describing how the brain changes with aging and in Alzheimer's disease and the tracts and parts of the brain that are affected. So focusing on the white matter at the bottom there. And then we talked about resistance training as an intervention. And looking at randomized controlled trials in those at risk of cognitive decline, you can see that resistance training, so lifting weights, preferentially protects the white matter, reverses white matter lesions, and prevents the atrophy of those white matter tracts. And there's a dose response. So at least 2 sessions a week for at least 6 months, and there's a number of ways or reasons why this might happen. It's a direct neuromuscular stimulus of the brain. We see improved function, and when you have improved function, then you go out and do more things, and then also improved metabolic health, which we know is really critical, which is metabolic health and metabolic syndrome are more common in those born preterm, and is also a significant risk factor for Alzheimer's disease and cognitive decline. So then when we think about all these risk factors, very similar to the number of therapies we might need to screen to find the best combinations, there are also a large range of risk factors for cognitive decline and dementia. But kind of promisingly, current estimates suggest that 40 to 70% or more of dementia is preventable. However, how all those risk factors interact is still really unknown. So I've just been recruited as a principal investigator of this citizen science project run by this dementia charity in the UK. So the Cognition Biobank, which is collecting data from tens, if not hundreds of thousands of individuals to look at how all these different lifestyle factors may affect their cognitive function. So the charity has this free, validated online cognitive function test that includes aspects of executive function, working memory, and processing speed. We currently have cognitive function scores from nearly 500,000 people, and then recently added a 144-item lifestyle questionnaire, so we have about 25,000 people who've answered that. And questions that we might ask are like, how well does lifestyle predict cognitive function, and can then we build and test models for how lifestyle affects cognition, and which are the most important predictors? So just some basic data from the database. We see that cognitive function on this test declines with age, as you might expect, although it sort of starts to accelerate slightly once people get into their 60s. And before I joined, they had what they called the Dementia Risk Index, which was using all these different questions in kind of this linear combination based on best guesses. And they just assumed that all the different questions in the questionnaire were similarly important for cognitive function. And so you can plot that risk index on the bottom against cognitive function score, and you get an R-squared of 0.04. So about 4% of cognitive function is predicted by this index, which isn't very good. And I was kind of a bit disappointed by that. So that was kind of the first project for me, was to see, well, can we improve this a little bit? So one simple way, and this is just the very beginning of this project. So running a simple machine learning model called a lasso to kind of figure out which are the most important responses out of all of these. And so this model was developed in 10,000 individuals and then tested in a held out portion of 5,000. And you can see, so this is in the validation data set on the left-hand side. You can see it's much better now. We can predict 25% of the variance in cognitive function just with this sort of very simple change to the model. So this is a great start. And then you might say, well, what were the most important things that we've seen so far? And so that's there on the right-hand side. And we still need to kind of figure out what all of these things mean, but some of the stuff that you might expect is there. So not smoking gives you a beneficial effect. Going out, so working from the right, if you don't go out frequently, and we know social interaction is important, that's associated with a lower score. The most important thing was in those who don't supplement with antioxidant supplements. And there's a number of reasons why this might be. So there's actually a lot of evidence now that in those who have a very low intake of dietary antioxidants, particularly polyphenols, they have an increased risk of dementia that you can reverse or cognitive decline. You can improve that if you supplement. So this is related to diet quality as well as the potential benefit of supplementation. And we know that oxidative stress is really important as a risk factor. However, different kinds of, the people who take supplements and the people who don't are different. So we also need to figure some of that out. But you'll see some common things that I've already talked about. So needing both arms to get out of a chair. So some aspect of frailty, we know that's a risk factor. Hearing loss is a significant risk factor. Regularly consuming refined foods, smoking more than 20 cigarettes a day. So all of these things, as you might expect, are important predictors. So then this is kind of the next step is we take all of these factors and we try and build, this is kind of a model for a statistical model that we hope to build for how all these different factors then affect cognitive function. So you can see these modifiable factors around the outside, they interact in different ways. Some of them are sort of stimulus specific. We talked about the importance of stimulus. Then there are some things that you might be able to measure as outputs of that system, like hormonal status, metabolic health, and then all of these things are then affecting the brain. But to kind of tie all of these things together, this is a more simplified version of the model, and it's designed, or kind of currently in this version, is for adults who are trying to prevent cognitive decline. But if you squint, I think it actually relates to all the different scenarios that we've talked about today. So the three S's are stimulus, supply, and support. Stimulus being the importance of stimulating function in the brain. We know that when we stimulate parts of the brain, it draws in more supply, so more we get neurovascular coupling and we get an increase in blood flow, so we need a healthy blood supply. We also need nutrients to build the neural substrate, so omega-3 fatty acids become important, choline and other structural components become important. And then if we're actually going to adapt, we need to sleep because that's when the adaptation occurs. But we also want to avoid things like stress that impair that process. So this is how it might work for an adult. But if we think about stimulus, we know that stimulus is really important for the extremely preterm brain. So, and the right kind of stimulus. So if we raise those infants in completely quiet environments, that's actually potentially detrimental. But music can be beneficial, as can tactile stimulus like kangaroo care. And we also know that the blood supply is really fragile, but also critical. As are omega-3 fatty acids and other aspects of nutrition like iron, really critical for the developing brain. So that, the supply makes sense there as well. And they also obviously need sleep. So this model kind of, I think, works across multiple different scenarios where we're trying to optimize the health of the brain. So in summary, first I told you about how neonatal neuroscience has this unique pipeline of developing therapies, and that's something that we're trying to improve with new models and drug screening platforms. But I will say that we still need to prove that the ferrets can translate to humans. If we're trying to predict outcome, I think we need to start zooming out rather than zooming in. And particularly if we're trying to intervene and improve cognitive function as that brain gets older, we need to do that at the organismal level rather than trying to tinker with the human source code at the cellular or biochemical level. But if we do that, I think there's lots of potential to intervene in everybody, but also particularly in those born preterm. Thanks to everybody here, everybody in the lab, Sunny Jewell and Elizabeth Nance, two major collaborators on all the work that we've done, as well as, of course, the funders who allow us to do this work. I'll just summarize with this. And I think we've got time for questions.
Wow, the whole lifespan. Thank you so much. If you are online and you have a question for us, please feel free to enter it in the chat. And if you're in the room and you have a question, raise your hand. I will bring you a microphone so everyone can hear. Hello, Dr. Brogan. You're the first one I saw.
Hi, thank you for that great talk. Since it is 2024 and everybody has one of these, is there any work done with that? Especially, I would imagine, with teenagers, but even old people like me.
Yeah, so I mean that would be a talk in its own right, but there are a few things that are potentially important. One is that if we think about social media and technology use, there's a very nice book that Jonathan Haidt just wrote called The Anxious Generation, which talks about how, so for anybody who's interested, I'd read that book. His work is really phenomenal, talking about how particularly the social media environment and technology is affecting adolescent brains and how that's changed in the last couple of decades. When we think about that for maybe people who are slightly older, there is some benefit from social media as well as this technology. We know that we can leverage technology to augment cognitive function. If you do like complex video games, maybe hard to do on the phone, but in other settings that have like 3D environments that you explore, those have been shown to give like transferable benefits to cognitive function, because again, you're just like, it's this novel complex stimulus that you're applying to the brain. Social media is also useful if you're connecting to other people in a meaningful way. However, there's been some very nice studies done looking at how social media affects how we internalize our social rank, and it becomes a social stressor when we spend all our time looking at other people who are happier, richer, better looking than we are. And that... You don't have to rub it in. Unfortunately, we all do this. So what this does is there's some nice work done by George Slavich and his colleagues at UCLA. This creates a social stress that activates an evolutionary conserved physiological response to social isolation. It's the same response. And then this drives sympathetic activation and a chronic inflammatory status. So how we're using the technology is really what matters. And the final piece being that constantly staring at a small screen, this process of foveating, again, can stimulate some sort of sympathetic stress drivers. So it's important to focus, and that's a part of focus, but then we also need to spend time not doing that. So time outside, time with a wider field of view, time in nature, all of these things can then offset some of that. So it's not that this stuff is inherently bad. Part of it is just that we never actually rest and recover from what is actually sort of quite a stressful mental work to do.
Tommy, that was such a great talk. Thank you. So a million questions. You could ask a million questions about what you presented, but I was curious, like on a personal level, because there's a lot of folks in the audience who are early in their careers, and I'm just wondering about your own path to what you're doing now.
Yeah.
Would you have expected when you started medical school that you were going to be in this line of work? And
No, not at all. And there's never been a plan, as you can probably see. And so there's a few things that have been kind of fundamental to some of this. One is, just fabulous mentors who've allowed me to do what I want to do. Like, I'm having Sunny as my division head, who's like, your job is to do interesting research and go and do interesting things. So as long as you're doing that, that's great. You're doing your job, which not a lot of institutions or bosses would necessarily let you do. And then kind of leading up to that, two main things. One is just always being open to a conversation and being nice to people. Somebody asks for help, somebody asks for information or support. That then leads to something 10 years down the line that you never expected. So that kind of building connections to people is really important, as well as, every step, going to medical school, doing a PhD, moving to the US, some of the companies that I've gotten involved in. It was all a spur of the moment. Somebody asked you, you're like, oh yeah, that sounds interesting, I'll do that. So that's kind of what's driven a lot of it. There's no real great advice in there because it kind of, some of it is maybe serendipitous, but I think those kind of building relationships with people has really helped.
Thank you so much for this really amazing talk and great, work, and so vast across the lifespan. I love that. And I guess I was really intrigued by the data you showed with the graduate school level education, maternal education, in comparison to the other levels. And I'm curious if you could comment a little more on how much of that is nature versus nurture, and if, tongue in cheek, but the intervention being, okay, everyone goes to graduate school. Obviously that won't happen, but where are the most important levers when you're looking at that data in terms of intervention, since you're really identifying that holistic level of neurocognitive kind of support for infancy that's so important?
Yeah, it's such a fantastic question. And in reality, when we do this kind of observational research, the variables that we collect are incredibly crude. So the maternal education becomes this catch-all for all these different things that might be part of that. I think that we have enough information to suggest that, yes, of course, each individual has a brain that works differently and is good at different things, right? That's an inherent part of humans. And actually, my friend Chantel Pratt, who's another faculty here, just published a book called The Neuroscience of You that talks about that. Like, every brain is different, and that's a good thing. So some of it is nature, of course. But I think that there's also lots of evidence to suggest that we have so much more capacity individually, and it's really dependent on the environment that it goes into. So maternal education as a proxy tells you about resources. It probably tells you about nutrition, which we know is important. It probably tells you about time and direct interaction, like how much. So say I know that socioeconomic status predicts how much like reading there is in the home, right, as a stimulus. And then it's also the absence of other chronic stresses that we know are an important part of that. So I can't tell you which is the most important. I think we're just kind of, hopefully over the next decade or so, going to start to build models that say, this is the most important intervention point. But I think that kind of proxy tells us that the environment is so critical, and that gives us the opportunity to intervene, even if we don't know all the exact fine details yet.
Thank you. A question online from Dr. Jewell. Fantastic talk. Thank you. I was wondering this question too. What antioxidants do you recommend? Also, feel free to give us your entire longevity regimen in the last 1 minute of this hour.
So specifically to the question, when you dig down into the other supplements and some of the research, vitamin E and vitamin C, or vitamin E and vitamin C, both seem to be important. Of course, you can get them from food. You don't need to supplement with them. From a supplement standpoint, there are two things that are probably best studied. One is blueberry anthocyanins, or anthocyanins from berries. And again, there are actually several studies showing that just giving people wild blueberries, like frozen wild blueberries you can get from the grocery store, significantly improves cognitive function if you take like 1 to 2 cups. So, and even if that doesn't, like, the worst case scenario is you just had some nice berries, right? It's sort of like super, super low risk. There are studies showing significant improvements in cognitive function with berries, cranberries, strawberries, and generally as powders. So those are the antioxidants that I think are best. Other carotenoids are really important as well. Lutein, zeaxanthin, which is sort of the yellows and reds that you can get from vegetables, and astaxanthin, which is what makes salmon pink. So maybe some local salmon. That'll probably do the trick.
Awesome. Wow. Thank you so much, Dr. Wood, and everyone for being here. We're all going to go to the gym and Whole Foods after this. Just a reminder for everyone who's here, thank you so much for coming. We are not here next week because our team has a conflict. There's a big ethics conference at the same time, but we'll see you all in 2 weeks. And thanks again, Dr. Wood. Really appreciate your help.